So, welcome to this class on Neuroscience of Human Movement. This is part 4 of the class on primary motor cortex. So, in today's class we will be talking about how populations of neurons contribute to activity or movements, right. In particular the work of Georgopoulos, where he showed that movement direction was encoded in a group or in a population of neurons. We will discuss the details of this work and how effects of level and direction of force that is exerted are represented or at least coded by neurons in the primary motor cortex. Okay. This is the work of suppose Ebfetz, I think. Okay. So, in the 1980s, Apostolos Georgopoulos and his colleagues did an experiment. Such experiments are still being performed, very popular paradigm. So, let me try and explain the paradigm first. This paradigm is called as the center out reaching task. So, what is this? Let me explain. So, there is a central home position. So, I am going to draw the central home position with the letter H here, and there were different targets. Okay. These different targets are, it could be to the east, okay, or it could be to the north, it could be to the west, it could be to the south, it could be of course to the northeast, to the northwest, to the southwest and to the southeast. So, effectively there were 8 directions, okay, and monkeys are trained to reach to these 8 directions. Okay. Such experiments are still being performed in monkeys and in humans currently. This kind of reaching task where you start from a central position and go out in a direction are called as center out reaching tasks. Very popular paradigm, hundreds of papers you could read that use these tasks to study how movements are controlled, right. So, they used a center out reaching task to wherein different directions, monkeys reach to different directions, and they recorded from a whole set of neurons, a whole bunch of neurons, right, not just a single neuron, a whole set of neurons. And what they found was that when you take combined activity of a population of neurons, then you could predict the movement direction. Okay. So, let me try and explain. On the left here on the left you see activity of a single neuron, right. So, this neuron is very active in that direction, which is the 135 degree direction and the 180 degree direction. You see that there is, so these are raster plots. So, the more the activity, more number of lines will be here and the darker will be the shades. So, for example, this and this, right. So, this neuron is very active in these 2 directions and slightly less active in the 90 degree direction, so or the 225 degree direction, right. So, in these 2 directions it is less active, and as you move in other directions its activity continues to reduce in directions exactly opposite to the most active directions, which are that one and that one. At in directions exactly opposite to these, which are, what are these 2 directions? That is the direction that is opposite to this one, and that is the direction that is opposite to that one. Let's name this for the sake of convenience. So, I am going to call this direction as the 180 degree direction, this as 315 degree direction, okay, does not matter. So, if you see 135 degree direction, this neuron is very active, and 180 degree direction it is very active, whereas in the 0 degree direction and in the 350 degree direction it is very silent. This is for a single neuron. What this shows is that movement direction or specific neurons are active depending on the particular direction in which the movement is happening. Specific neurons have some preferred directions in which they are most active, and in other directions they are less and less active, and in directions exactly opposite to their preferred directions they are very silent, right. As you can see, there is a lot of white space here and here, is it not? From this we could deduce that that these are the least preferred directions for this particular neuron. From this data we could come up with a directional tuning curve for this neuron. So, this tuning curve will look something like this. So, something like this with its highest magnitude for example at 135 degree direction. At 135 degrees this is going to have the maximum amplitude, and at at two other directions that could be 180 degrees, this is 180 degrees, and 90 degrees, the amplitude is slightly less. At other directions the amplitude is going to be much lower. This is for a single neuron, but note there may be other neurons whose directional tuning curves. So, this is called as the directional tuning curve for this particular neuron. Okay. What Georgopoulos did was that he found directional tuning curves, similar directional tuning curves for a whole bunch of other neurons, and he found that different neurons had different preferred directions. So, effectively what I can do is that each neuron's activity in a particular direction, if I represent using a vector, then I add the activity of all the cells together, or I vector sum over each direction, then what I will get is that the entire population of neurons will predict the overall direction made by the primate, or in other words. So, what happens is that if I do this for a whole bunch of neurons, there may be other neuron for which, for example, a similar directional tuning curve will be there. For that the maximum amplitude will be at 90 degrees. There will be another neuron for which a similar directional tuning curve will be there, for which the maximum amplitude will be at 0 degrees, right. And there will be another neuron for which one more directional tuning curve will be there, for which the maximum amplitude will be 315 degrees, like that. Now, note when the monkey is moving in a particular direction, I know the direction in which the monkey has moved, and now if I add the activity or vector sum the activity of all these neurons, then I am going to get a response like this. So, each line here represents the activity of represents the direction of the activity of a neuron, and when I vector sum all this I am going to get a direction like that. For example, this is the vector sum of all these lines. Similarly, the line in the center, that line is the vector sum of all these other lines. This line is the vector sum of all these other lines, and so on and so forth. From these neurons we are able to predict, from these activities or from this approach I am able to predict what the direction of movement is from the activity of neurons. So, from this we could deduce that a population of neurons encode direction of movement. Note importantly that movement direction in this case, right, is not specifically controlled by a single muscle. Note, depending on the we will have to go back to our earlier classes and remind ourselves of how this is done. This is a center out reaching task depending on the direction of movement. So, that means if this is the center, suppose the place where I am holding, or the place where I am holding this remote is the center, let us assume that. Then if I am doing that, right, that is one moment, and if I am doing that, that is another moment, if I am doing this, that is another moment, that is another moment. Note, fundamentally there is a huge difference in the way these movements are executed, right, because the muscles responsible for that movement is different from the muscle that is responsible for the opposite movement, is it not? Because these two, one is flexion movement, the other is an extension movement. So, flexors are involved in one case, extensors are involved in other case. So, individual muscle activity is not considered here, but rather the movement direction itself is considered. So, that means coordinated muscle activity, output movement is the output of coordinated muscle activity. So, coordinated muscle activity is predicted by a population of neurons. This approach to using vectors to study populations of neurons has become popular, very popular in neuroscience. This is called as the population vector approach. Okay, this was championed, first pioneered by Apostolos Georgopoulos and his colleagues. So, this is seminal papers of 1982 and 1983, right, showing how depending on the movement direction the activity of different sets of neurons varied. Okay. And note importantly that one is that movement is what is encoded, not individual muscle activity. Note also how this varies in contrast with what we saw in yesterday's class, in the previous class. In the previous class what we saw was that Edwards showed that activity of specific muscles are encoded by individual neurons. The fundamental difference between that approach and this approach is that in this case multiple neurons are recorded and their population activity is what is being used to predict the movements. Whereas in the previous case, in Edwards case, only a few neurons or only a single neuronal activity is used to predict correlations with muscle activity and correlations with kinematics and kinetics of movement. Okay, fundamental difference in the approach. Okay, here, so what we have learned from this slide is that we can use activity of a population of neurons to predict movement direction. Okay, what was also shown by Epfetz and his colleagues. So, this is work of Epfetz in the 1970s and 1980s, late 1970s and early 1980s. Okay, is that the level and direction of force exerted coded by the neurons in the primary motor cortex. Especially the following is true. Okay, so here different lines that I am going to draw represent activity of different neurons. Okay. So, this is torque, I suppose this is firing frequency of different neurons. Each line that I am going to draw represents the activity of one neuron. So, if you take one particular neuron, as the torque that needs to be produced increases, the firing frequency increases. By the way, what is this task? So, in the previous case we saw this is a center out reaching task. In the previous case, in the case of Georgopoulos, that is a center out reaching task. So, there is a center here, you have to reach to different directions, right. This is a center out reaching task. But what did Epfetz do? In this case, the task is static force production or isometric force production. What does this mean? This means there is no real movement. There is an object here and I am trying to hold that object and produce force on that object in multiple directions and at multiple levels. Right. And this object is not going to move, it is a strong object that is completely fixed to the to a table or something like that. So, I am trying to work very hard, right. I am trying to push it in a particular direction. I could increase it to a level, I could decrease it, I could push it in the opposite direction, I could do a whole bunch of things, but this is not going to move. So, this is isometric force production, that involves no real movements. So, in this case the torque that is produced, or the muscle torque that is produced, is varying linearly with firing frequency, or as the torque increases, the activity of this particular neuron or the firing frequency in this particular neuron increases. So, let us take a different neuron. In the case of a different neuron, the torque increases like this, in a different neuron it increases like that, in a different neuron it increases like that, in a different neuron it increases like that, not like that, like that, in a different neuron it increases like that, and so on and so forth. These are activities of say 5, 6 neurons, right. Like this we could record activity of multiple neurons. So, multiple neurons, obviously the slopes of all these lines will be different, but what is important to note is this message, that in general this is true. If I take the average of all these things, approximately this is the line that I am going to get. What is the message from this? The message is that if I have to increase the force, then the firing frequency will increase. Importantly, what this means is, in the case of isometric force production, this is true in the case of isometric force production. So, this provides crucial, very strong evidence in support of the hypothesis that in the absence of movements, when there is no movement, there is no such thing as kinematics. This is isometric force production. In the absence of movements, force level is encoded by the primary motor cortex. So, the idea that force is being controlled is a control variable. Some evidence, at least in the range of forces considered and the firing frequencies considered, in the range of forces considered and in the set of neurons taken, at least you could say that there is a very nice correlation between force level and the activity of the primary motor cortex neuron. So, in summary, what we have seen in this class is the seminal work of Apostolus Georgopoulos in showing that the direction of movement is encoded by a population of neurons. So, using the famous population vector technique, okay, and the work of Epworth, where he showed that the level of force is encoded by the activity of primary motor cortex neurons. Okay. So, we will stop here and we will continue these discussions in future classes. So, thank you very much.